Well, before we get started, I wanted to make a couple announcements. Hi, everybody, and welcome to our May seminar. I'm Kathleen Kay. I'm the current CalBotSoc president, and I wanted to remind everyone that we have student research awards that are open right now. The proposals are due May 15th, which is coming up in less than a week. And I want to ask you to encourage your students to apply. We have a new streamlined application procedure. You can find all the details on the website, but it should be more straightforward and easier to access than it has been before. So please apply or tell your students to apply. And also just I want to encourage you all to send your manuscripts to Madronio. We have a new team of associate editors and copy editors on board to process manuscripts faster, and we're looking for good new manuscripts. So if you have something interesting about the Western American flora, consider sending it to Madronio. Okay, I will stop my shameless plugs and just tell you that it's my great pleasure to introduce Mike Simpson as our speaker tonight. Mike probably needs no introduction for most of you. He has been a longtime member and supporter of the Botanical Society and also professor at San Diego State University, where he's now emeritus. He is a curator at the San Diego State Herbarium, where he's currently located, and also the author of the Plant Systematics textbook, which objectively speaking has the nicest images of any systematics textbook, and it's the best one if you want to show your students the key characters of any family. He has longtime interests in plant taxonomy, systematics, floristics, with special focus on the popcorn flowers, which he's going to talk about tonight. Also the Hemidoraceae, which we got a little taste of last month at the annual banquet. So Mike just spoke at our annual banquet in Fullerton less than a month ago, and gave a really fascinating talk on a lot of the things that he's worked on over his career. But I know that many of you were not able to be at the banquet and see his talk, so he was very gracious to agree to give a talk tonight for our seminar series, where he's going to take more of a deep dive into his work on the popcorn flowers and the amphitropical distributions, where species or genera are spanning the neotropics, which has always been a very curious pattern. So welcome to everyone, and I'll let Mike get started with his talk, and we'll have questions afterwards. And please mute yourself if you haven't done so already. All right, Mike.
Okay. All right, you see the slide okay, Kathleen? I hope so. Okay, I'm assuming everything is okay. As usual, I can't hear anybody else. But welcome, everybody. I gave part of this talk at the CalBotSoc banquet. I wanted to focus tonight on this topic, this interesting pattern that has been observed for over 100 years. And this is a pattern that was first discussed by Asa Gray and Joseph Hooker. And this is the pattern seen in this map, in which plants occur both in Western North America and Western South America, primarily, the two continents. This is the original publication of Asa Gray and Hooker in 1880, and they were looking at the vegetation of Rocky Mountain plants and noted that some of them occur also in South America. So they started this, and subsequently other authors have published in this field. I wanted to emphasize Raven in 1963. I'll talk about that publication in a bit. And then because of my interest in this in 2017, I'll talk about a publication I and three co-authors put out, kind of a reinvestigation of this phenomenon. So this is the topic, the main topic. I'm going to touch a little bit on the popcorn flowers because they're so well represented in this phenomenon. And it's called the American amphitropical disjunct phenomenon, you could say, or events, AADs, we refer to them, which I always joke is not a disease or a genetic syndrome, but refers to this interesting plant distribution pattern. So, of course, we're talking about the Americas, the Western Hemisphere, North and South America. Amphitropic, referring to either side of the tropics, north of the Tropic of Cancer, south of the Tropic of Capricorn, and disjunct, of course, referring to populations of plants that are well separated from one another beyond their normal distribution. So this shows that this is a map modified from the reference below Davis and Holmgren, one of several global ecological zones. And you can sort of see at a glance the tropical areas largely in purple there, with temperate, desert, and boreal regions on either side. So that's the amphitropical zone. In the dashed red line, you can see what have been defined by various authors as the boreal zones that I'll talk about a little bit later. So I did want to mention that in this phenomenon, there are a number of moss species that have been recognized to have the AAD pattern, distribution pattern, 66 listed there. Several 24 liverworts, and many lichens exhibit this pattern as well. So I'm not going to be talking about those groups. I'll be focusing on the vascular plants. And as I mentioned, Raven was really the first one in 1963. He put out a wonderful article, what we could call a compendium of AAD vascular plants, with listing 124 examples. And of these examples, he cited plants that were the same species, or what he called congeneric species, considered to be each other's closest relative or very close to one another. And this was pre-molecular phylogenetic analyses, so he did what he could at that time. And in 2016, we organized a symposium at the botany meetings in Savannah, Georgia, that resulted in this symposium volume in AJB in 2017. And the article I'm going to be talking about is this one that I headed out with Lee Johnson, Tamara Villaverde, and Matt Williams, which was updating Raven's work really and talking about some aspects of AADs, some interesting phenomena about them, and then some future directions that I'll try to summarize at the end. Okay, and in this compendium, we talked about what amphitropic meant, and we actually categorized it in different ways. Of course, we didn't include plants that had a continuous range, but I wanted to mention this. We only considered, of course, plants that are native to North and South America in this pattern, not those that were naturalized. And that in itself is an interesting phenomenon, talking about this same pattern of distribution with naturalized plants. But what's interesting, this is not always an easy thing to determine whether something is native or naturalized. For example, this is a Schultzia californica, and I always ask the audience, is it native or naturalized? Of course, they'll say it's native to California, but these photographs were taken in Chile. So it is naturalized in Chile. So that's just a current example of a plant which we know is naturalized from recent historical information. But in other cases, it's not always clear. When was it introduced? It's a detective story sometimes to determine if something is native or naturalized. And sometimes there were competing hypotheses. So we did the best we could and cited where there were discrepancies. In the great majority of cases, we were certain they were native in both places. So how did this distribution pattern occur, this amphitropic disjunct pattern? And we think the most likely explanation is long-distance dispersal by birds. This is accepted by most people in the field. A lot of articles have been written on it. It's still controversial, but this is what I think and most people think was the explanation for how they got from one continent to another. And there have been plant fragments found on the feathers and feet of birds when birds were captured and examined for plant fragments. So this is certainly possible. And it's interesting that you may not be aware of this, but this Collins wrote his thesis at what was then Rancho Santa Ana Botanic Garden on, you can see the title, Mechanisms for Long Distance Dispersal, in which he empirically examined propagules from species that had an amphitropic distribution, talked about bird migratory patterns, and actually determined empirically if these plant propagules could attach actually to the feathers of birds. He actually did empirical work on this and concluded that it was most likely epizoochory on the outside of birds that these propagules were attached. And again, these migratory patterns, long distance flyways of birds that I'll mention again later in the talk. So this is our calculation by family of the number of AAD events. We came up with 237 at the time, and I'm in the process of revising this. Another 10 or so can be added to that list. And you can see that the grass family, the Poaceae, is the family that has the most representatives, which is you would expect grasses get around well. But I wanted to point out that the Boraginaceae with 19 AAD events, and I'll talk about why I call them events a little bit later, is well represented. And of those 19 Boraginaceae, 18 of them, as the family is defined now, is in our popcorn flower group. So I'll diverge a bit on the popcorn flowers. You can see the composites and legumes are well represented with 16 each, the sedges with 15, and the polymones with 11. So these are the top 6 families that show this AAD distribution pattern. Now, since popcorn flowers are so well represented, and since I studied, I just want to diverge a little bit and introduce you to them, because I'll be talking about examples from the popcorn flowers. I considered a fascinating group of plants to study. We now call this group collectively, which is technically the subtribe Amsinckiinae. We're using the common name popcorn flower to refer to the entire subtribe in our case. And this is a slide from Wayne's World that shows you why they're called popcorn flowers. Somebody thought the clusters of white flowers in the field resembled popcorn. So that's the origin of the name. I wanted to point out that popcorn flowers, as you'll see, occur in North and South America. This is one photographed in the Andes Mountains of South America. This particular species, Cryptantha allosoides, it's very fragrant. We don't usually associate popcorn flowers with a fragrant odor, but some of them can be, particularly in some of these South American taxa. And just as a review, I think many of you know this. This is the popcorn flowers are in the Boraginaceae, one subtribe of the Boraginaceae. All Boraginaceae, as we define it now, have this four-lobed ovary that you can see there. Let me get my, I don't know if I can, but anyway, if you can see my cursor, there's a four-lobed ovary of Borrego. And each of those lobes typically develops into a nutlet containing a single seed. Now, in the Amsinckiinae, the popcorn flowers, the receptacle is elongated to form what we call the gynobase, to which the nutlets are attached, each of the fruit units being called a little nutlet. And then there's a style and stigma as well. And all these characters can be important in identification. Note the calyx surrounding the nutlets, and that calyx is often very bristly. The nutlets attached to the calyx can be in itself a dispersal unit. So I got interested in popcorn flowers because of the interesting nutlet morphology, and traditionally things like how they were attached, what's called the attachment scar, has been used to characterize or delimit various genera within the Boraginaceae, within the popcorn flowers. And for example, many of you know that Cryptantha, as it had been defined, has this long slit-like attachment scar called a ventral groove, whereas Plagiobothrys has what's called a ventral ridge above the attachment scar. So that's how those two genera had been defined in the past. I'll just mention this, just the most recent article we published with my former grad students, now three of which we went on to get PhDs, Matt, Kristen, and Mackenzie, and then Lee Rittman was very instrumental in this article as well. We published an analysis, molecular phylogenetic analysis using genome skimming. So we got virtually all of the chloroplast DNA, a lot of the cistron, a lot of the mitochondrial DNA, and published this cladogram at the time. And I just wanted to review it real quickly just to point out that of these members of the popcorn flowers, the genera are indicated there as we define them today. I'm going to just show you real quickly the ones that do show an amphitropic distribution. And Cryptantha SS, as it's explained, as it's labeled here, has the most members that have an A distribution. This is the genus that I have learned to love, I could say. It's one of those groups I've become pretty familiar with now. And this shows the variation in nutlet morphology. And a lot of you know that the size, shape, sculpturing patterns, and attachment scars, and the number of nutlets per fruit can be used to classify both Cryptantha and other members of the group. So Cryptantha are well represented as members of the popcorn flowers that have the AAD pattern. Johnstonella and Greeniaceae are two genera that we pulled out of Cryptantha. They were within the genus Cryptantha until the article in 2012 by Kristen and I. And this is now in the Jepson flora. They've now been separated. They fall out well separate from Cryptantha SS. And these two genera, in addition to two others, Oriocaria and Aremocaria, as shown above, used to be Cryptanthas, but now are separated out. Johnstonella and Greeniaceae have AAD examples. So they have members of those two genera that occur in South America. Here's Greeniaceae, which has these typical red roots and bracts. It's found in South America, and several species of Johnstonella, real interesting genus that we separated for Cryptantha, occur in South America as well. Another genus in South America in our group is Amsinckia. Many of you know Amsinckia, the fiddlenecks, as they're seen here. They have, interestingly, these bilobed cotyledons, but they're other than some Oriocaria, they're the only popcorn flowers with really orange or yellow flowers. So they're a very distinctive group that occur in South America. Pectocaria, seen up above, occurs in South America. These are the comb seeds. They have these distinctive fruits with the nutlets are in fours always and diverge. They're in one plane, roughly speaking, and have interesting morphology. Several species occur in South America as well. And then Plagiobothrys, a lot of species of Plagiobothrys occur in South America. Here are some of the nutlets of Plagiobothrys from our high resolution microscope that you can see the variation again in morphology. I wanted to point out this last little clade. We call it the Meridimae clade. We haven't separated it from Cryptantha as of yet because we have a relatively weak backbone in our analysis. It's unclear whether it belongs in the genus Cryptantha or should be treated as a separate genus. Just pointed out, they look like Cryptanthas, and there's several species. I think they're upwards of 17 species that are going to belong to this clade, a separate clade. And one of them down below is Nisocaryum stylosum, which has been treated in a separate genus in the past. And I talked about this at the banquet in a little more length, but just wanted to mention it, that we now have evidence that this thing that occurs 900 kilometers off the coast of Chile, seen in the upper left, on a very isolated and rugged island, Isla San Ambrosio, is probably going to be transferred to Cryptantha. We have pretty good molecular data for that, and you can see the nutlets to the lower right have the typical ventral groove of Cryptantha. So those are the players in the popcorn flowers that show this American amphitropic disjunct pattern. So to go through taxonomic groups, we separated them by, again, these broad groups in our paper, and the majority of examples of AADs, 57%, 135 examples, are the same species. So illustrated here in Ambrosia camasonis, I'm going to show you a few examples you may be familiar with, Californian plants. And I want you to think about this. We're calling this the same species, of course, and that's based on morphology. And this has been traditionally done. It would be a matter of having an herbarium sheet of a Chilean Ambrosia camasonis and a Californian one side by side. You can't tell them apart morphologically. But as you can gather, a lot of these have been separated for a long time, generally millions of years, and have been reproductively isolated. So calling them the same species is just a matter of convenience. They are morphologically similar, but in terms of reproductive isolation, they've undoubtedly been reproductively isolated for a long time. But you could cross them in a controlled environment in general. And Raven actually did some of that work. Some of the other examples that you may be familiar with that are considered to be the same species, Xylocarpus brevissimus, largely vernal pool species that you may be familiar with in California. Lepidium nitidum in the Boraginaceae, little lepidium in our coastal sage scrub. And Senicula crassicalis, which you may know in the Apiaceae. And here's one that we have an indication as to when it diverged, about 1 million years ago. And this is, as I'll point out, the timing of divergence, I'll talk about a little bit, is based on molecular phylogenetic analyses where we have some knowledge of divergence times. And these are fairly limited, but where we did have that information, we could estimate the time of divergence. And as we'll talk about, the direction of dispersal. Okay, so those are some examples of the same species of plants that occur on both continents. And then we looked at infraspecies. So these are varieties or subspecies, of course, that occur on the two continents, North and South America. In some cases, they were the same infraspecies. In other cases, different infraspecies. So we did separate those two. We came up with about 17 examples, about 7% of the total. Here's an example. Our Greenococcus that I showed you earlier, and our popcorn flower is an example of an infraspecies. It has two varieties in North America. Variety circumcissa by far is the most widely distributed variety. You can see it in Western North America, and there are a few populations in Argentina of this variety, the same variety. Again, they're indistinguishable morphologically. There's one other variety in California that's very rare, rosulated, that occurs at high elevation in the Sierras. Cryptantha marittima, real interesting species of Cryptantha, is another example of an AAD that has different varieties. There are actually 4 varieties. John Rubin and I published on this recently, and kind of reinstated variety pilosa, which had been merged with other varieties. But the variety pilosa is found both in North America and in South America. Quite rare in Argentina, but it is present, we think, a natural native population. Plagiobothrys cholinus is an example of an AAD in our popcorn flower group. Many of you may know this. It's a common member of the spring flora in chaparral and coastal sage scrub habitats. And it has 5 varieties that are recognized now in North America and 1 in South America. And the one in South America is considered separate. In fact, that is the type for the species, variety cholinus. This used to be called Plagiobothrys californicus. Some of you may remember that, but it was realized it was the same thing in South America, and Plagiobothrys cholinus there had priority of nomenclature. So that's an example of a variety that is different in South America. Okay, so those were infraspecies. We also looked at species pairs, pairs of species that were each other's closest relative. And we evaluated this largely based on, again, molecular phylogenetic analyses where we had evidence that they were each other's closest relative. In a few cases, it was based on morphology where we thought that they were morphologically similar enough that they were each other's closest relative. So here's one example of that, Johnstonella of our popcorn flower group that I mentioned. This is Johnstonella angelica, which occurs largely in Baja California. And from our molecular analyses, it turned out to be sister to Johnstonella parviflora. And if you look at the picture of parviflora in the upper right, you can see the nutlet morphology of the two is rather similar, and there's some differences. But when I first saw Johnstonella parviflora, I thought, wow, this looks just like Johnstonella angustifolia, what we're calling Johnstonella angustifolia today. And it's actually most closely related to Angelica, Baja California. Just as an aside, I wanted to mention that fairly recently, some students in a Jepson workshop I was giving in Borrego Springs pointed out a plant that we collected, and it turned out to be Johnstonella angelica of this species pair. It was a county, state, and country record 240 miles north of what was thought to be the Johnstonella angelica in Baja California. And very recently, I'll point out that, don't mean to steal his thunder, but John Redmond discovered this in the Clark Dry Lake area of Borrego Springs, of Anza Borrego Desert State Park, rather, in a much more native habitat. So we're now convinced that it occurs natively in the United States, California, this species. So we have evidence that this is an example, again, of a species pair, that there was a dispersal event over 3 million years ago from North America to South America. As I'll talk about later, the direction of dispersal can be evaluated again from, as you may gather, from molecular phylogenetic analyses. So this is one we have data for. Another example of a species pair is Laria. This shows our Laria tridentata. And again, about 23% of the total examples of AADs are species pairs, nearly a fifth. But you may know there's a species of Laria, actually more than one species of Laria in South America. When I first saw Laria divaricata in South America, I thought, wow, it looks just like tridentata. In fact, the two had been treated as subspecies of one another, but were separated out most recently. And this one we have evidence for dispersing about 6 million years ago, and from south to north. Talk about that direction of dispersal pattern a little bit later. So again, an example of pairs of species that show this AAD pattern of distribution. And lastly, kind of uniquely, we were able to investigate clades of species that show the AAD pattern. And this had not been done by Raven. It was pre-molecular or pre-phylogenetic analyses. But here we looked at examples that showed either two clades or one species sister to a clade. So two sister clades or one species sister to a clade of species that showed this distribution pattern. And fully 1/5 of them, 20%, had this clade phenomenon. And as an example, again, in our popcorn flower analysis, here's an example. Here in the blue are North American members of popcorn flowers, and the orange are South American members. And here you can see an example of two clades that are sister to one another. And we can, of course, evaluate when those two clades diverge. And we can evaluate the direction of dispersal. You can see that everything ancestral to those two clades is in North America. So that's the most, we could say, parsimonious explanation that it diverged from North to South America. Okay, so those are the categories that we started out with, kind of taxonomic categories. And when you plot out the mean timing of dispersal of these different groups by taxonomic group, you can see some patterns. Now, there's quite a bit of variance, particularly the ones to the left, but obviously the ones that are the same species, conspecies and infraspecies, have a much more recent time of dispersal, of divergence, than the species pairs and the clade examples. But again, to the left, you can see there is quite a bit of variance, but the orange diamond-shaped figures point out the means. By no means is this statistically significant. We just plotted out what we had. Second, there we go. Okay, so here is a graph just showing those mean divergence times, conspecific, infraspecific, species pairs, and species groups. And you can think about this, and it makes total sense that the longer there has been separation between two members, to the lineages, we could say, resulting from a dispersal event, the more probability there is for them to diverge. So here's the dispersal event resulting in what we're calling today lineages, but on the two continents that we call the same species. Here is the slightly older in time divergence of infraspecies. With 3 million years ago, the average time for species pairs, you have enough time for one or both lineages to diverge in what we're calling separate species. And then with clade examples, the average being a little over 6 million years ago, there is enough time for those lineages to diverge, either one or both lineages into clades of species. So, the further back time of divergence times as you consider taxonomic groups makes total sense. That the further back in time they dispersed and diverged, the more probability that there would be for divergence and speciation into clades or into separate species. We also looked, and this is something that Raven had done as well, these bioregions, bipolar, which is not disease, it's referring to the two poles, Arctic and Antarctic regions, temperate, which includes our Mediterranean climate regions, and then desert regions. Some of these bioregions were not clear cut. So we did the best we could there. Some of them probably need to be evaluated again. But when you compare those three bioregions in terms of divergence times, you can see the bipolar comparisons have a much later, more recent mean divergence time than temperate and desert, and a much less variance than the other two groups. So thinking about this, if you here are the actual figures. So the bipolar examples are many fewer, 27 examples, as opposed to temperate 158 and desert 52, you can see. But the mean divergence times much more recent in time for bipolar than temperate and desert. And also look at the percentage of perennials versus annuals in the three groups. Virtually all of the bipolar plants are perennials. About half of the temperate plants are perennials. More than half are annuals. Of course, the annual plant duration has been very successful, particularly Mediterranean plants and in desert plants. But many more of the desert plants are perennial. So there's a trend there. We can't really explain, the bipolar plants, in general, plants that are at high elevation or in colder climates tend to be perennials. That's been discussed in the literature. Perennial plants can survive cold extremes, snow cover better than annuals, it's thought. So that's probably a function of their habitat. Now, the timing of divergence of the bipolar plants being much more recent makes sense, I think, in terms of thinking about what these regions looked like in the past. Of course, both the Antarctic and the Arctic regions were covered by ice and glaciers until relatively recently. In fact, there was undoubtedly some migration, of course, from the south in the Arctic and from the north in the Antarctic. So it certainly makes sense that they would have a relatively recent mean divergence time and very little variance. Dispersal direction is kind of interesting. This was noted by Raven, but we confirmed it is overwhelmingly from North America to South America, 81% of the total. So that's interesting to think about. But when you break it down into groups, all of the bipolar plants go from north to south. The great majority of the temperate, so-called temperate plants from north to south, but about half of the desert plants go from south to north, the other half from north to south. So interesting trends there that we can see based on these three bioregion categories. And we think that this is probably due to bird migratory patterns. I don't have a great deal of knowledge about the specifics of this, and it hasn't really been worked out, as far as I know, particularly for the desert plants in terms of evaluating the bird migratory patterns that might have been used in the past. Of course, bird migratory patterns might have been quite different in the past, but this is based on what we see in the present. With regard to the bipolar plants, though, we think the fact that there is you can see all of them dispersed from North to South America. If you look at the areas of the Arctic versus the Antarctic regions, those dotted dashed lines rather, there's a much greater area in the Arctic than in the Antarctic, and it may be more of a filtering mechanism. There would be a much greater probability, aside from bird migratory patterns, a much greater area in the Arctic might account for the fact that all of the direction and bipolar plants went from north to south. Okay, so here's an example of a desert plant. It showed a few examples of going from north to south, but as I mentioned before, Laria tridentata resulted from a dispersal event about 6 million years ago from South America to North America. Another example, is this is work done by Beryl Simpson and workers, Amsinckia. They came up with 4 examples of AD events, all of them again from south to north. And this shows the one to the right, Amsinckia glauca, which is a single species. In this case, others were clade examples. In the popcorn flowers, why I'm highlighting this, I mentioned that 18 of the 19 Boraginaceae are popcorn flowers. We actually wrote a separate article on the biogeographic history of the popcorn flowers. And based on the data we had at the time, came up with 18 dispersal events, all of them from North America to South America. I have 17 to 18 because one was equivocal. So 18 times popcorn flowers dispersed from North America to South America. So by far, for the size of the group, this has the greatest number of examples of the AAD pattern. And the timing of dispersal can vary tremendously, not just in popcorn flowers, but in all of these. The most recent popcorn flowers dispersal events occurred about 300,000 years ago. And the ones earliest diverging would be about 17 million years ago. And these are rough estimates based on the data we have, but you can see that this has been occurring both in the popcorn flowers and in other groups for a long, long time at various times in the past. For the popcorn flowers, we just plotted this out, and I think you can see, again, the rough correlation. The ones to the left tend to be the same species or infraspecies, have a much more recent dispersal time, and the ones to the right tend to be more clade examples in general, clade examples or species clade examples. So we see that same general pattern that we saw for all of the American amphitropic disjuncts. Okay, so in our article, we talked a little bit about what happens after the dispersal event. And, I always say the 2 take-homes are, 1, that plants can get around, and 2, when they do get around to a new environment, they can change. So what happens after plant dispersal? One of the things we noted was that when an AD event occurs, and when you see a shift from an annual to a perennial plant duration, it's almost without exception that shift to the perennial plant duration occurs in the recipient or the new continent. So in the new environment, we not commonly, but when it does happen, it happens in the new continent. You see a shift from an annual to a perennial plant duration. And it's been thought, this has been discussed, what would be the selective pressure? One possible explanation could be the environment that they occur in. For example, some of these perennial Cryptanthas occur in, again, high elevation areas. But it could also be that perennials would persist year after year. Most likely, a single propagule got to the recipient continent, most commonly South America, one single plant. And it has to survive for beyond that first season to make it, otherwise it goes extinct. So selective pressure for perennial duration might be advantageous because now you have a plant that can survive on its own and over multiple generations can produce propagules rather than being so-called forced to reproduce sexually anyway in a single year. Now, it should be pointed out that virtually, as far as we know, AAD plants are all self-compatible. So they are capable of pollinating, self-pollinating. This is called Baker's rule. That was been noted a long time. So this shows that in Cryptantha, there are several Cryptantha that are perennial. Now, all Cryptantha in North America are annuals, but there's several in South America that are perennials. This is one, Cryptantha capituliflora, photographed by my former grad student, McKinsey Mabry. And it looks a lot like what is now in the genus Oreochary, if you know that genus, which occur at high elevation in North America. They're all perennials. And this is why Johnston, the guru of the group, wanted to combine them. He made a mistake. he concluded that the perennial duration was ancestral in Cryptantha. Anyway, this is one example where a shift to the perennial duration occurred in South America in Cryptantha. And here you can see it on the cladogram that McKinsey and I published in a separate article showing this, the second blue square from the bottom are all South American Cryptanthas, and the black dot referred to perennials within that group. Another thing that we noted was this shift from chasmogamy to cleistogamy. You guys probably know what cleistogamy is, the phenomenon where flowers, some flowers, at least the cleistogamous ones, don't open up. The perianth remains at a kind of juvenile stage, whereas the sexual parts mature. And so self-pollination is obligate in a cleistogamous flower. And the thought is that maybe this is advantageous. It would better ensure that there is self-pollination in the plant that occurred in the recipient region, the recipient continent. There may not be pollinators. There would normally not be the same pollinators that would be present in the source continent. So cleistogamy may have been selected for. There may have been selected pressures for that. We do see this in Cryptantha. There are no cleistogamous Cryptantha in North America, but in South America, you see cleistogamy illustrated in the green and black in that South American clade. In fact, this is the type species for Cryptantha is a South American plant, Cryptantha glomerata. And I've seen this many times now. It's actually not uncommon in South America. And they do have chasmogamous flowers in the upper part of the plant, but they also have in the axils of the lower leaves, buds, flower buds that are cleistogamous and will produce normal types of nutlets, but they are, again, obligately self-pollinated. Some Cryptanthas have evolved these weird types of cleistogamous flowers that occur at the base of the plant, in addition to chasmogamous flowers above, but the ones that are down below in a few species can actually be pulled underground. Their nutlets look very strange. They're much larger than the nutlets above. This is sort of a secondary evolution, and the term cleistogene has been given for these cleistogamous flowers in Cryptantha. So it's kind of a specialized type of cleistogamous flowers that form larger nutlets that can be pulled underground by contractile roots. Interesting phenomenon. A third thing that has occurred in many AADs is polyploidy, whole genome duplication, and where you do see a shift from diploidy to polyploidy in these AAD plants, inevitably the polyploidy evolves in the recipient continent again. So we have these things happening in the new place, the new home for the dispersal event. And in Cryptantha, as an example, all Cryptantha of North America are diploid, but there are several Cryptantha in South America that are polyploid. In fact, the red asterisks there refer to hexaploid Cryptantha found in South America, and one in the lower South American clade. One of them is hexaploid, has been counted as well. So that's an interesting phenomenon, a polyploidy evolving in the recipient continent. Now, another interesting example of polyploidy you may have heard about is in Laria. We suspect that Laria tridentata, again, got to North America from a single dispersal event from South America, probably in the eastern part of its range, where all of the Laria tridentata are diploids. And this is work that's been done by Laporte and others. And as Laria migrated to the west, it shifted to polyploid. Now, this wasn't a direct result of an AAD event, but polyploidy nonetheless occurred in the recipient continent. Most of the populations in southwestern Arizona are tetraploid. And then furthest west in Arizona and most of the ones in southeastern California are hexaploid. So that's an interesting phenomenon where you have three ploidy levels of Laria tridentata. Laporte has done further work as an aside into the ecological, physiological significance of this. He's come up with virtually no correlations, in terms of habitat and moisture regimes and so forth. I suspect that there may be a difference in the fact that the ones that are hexaploid are better adapted to a drier environment. One of my colleagues may be investigating heat shock proteins in Laria and to see if there are increased heat shock proteins which are involved in protecting DNA in plants that occur in dry and hot environments. So that's another example of polyploidy in the recipient continent, Laria tridentata. We also noted changes where a few studies have been done evaluating changes in the rate of species diversification in AAD plants, and where you do see an increase in the species diversification rate, it's always in the recipient continent. So here's one example, an astragalus with many representatives in South America. This study by Shearson et al. Showed that two clades that showed exhibited increased rates of speciation were both South American. And that's where so this may be an example of adaptive radiation, where there's a release when a new propagule makes it to a new environment that's been shown in island biogeography. There's a release of competition, new niches available, and so perhaps an increased opportunity also, new pollinators, or no more competitors, no more competition from plants. And so this is a case where speciation rate may have been increased due to adaptive radiation in those groups. So those are some of the main players in AADs based on taxonomic group, based on bioregions, and the correlations that we saw in terms of what changes occurred in the recipient continent was quite interesting. So this is, in one sense, our analyses were somewhat rough. We did the best we could with the data that we have. We saw some interesting trends, but next steps, we need more molecular phylogenetic studies. We had only 72 of those 237 examples that had the molecular phylogenetic studies where we could clearly evaluate divergence times and direction. So we need some of you people out there, including some of you grad students, to do some of these analyses of AAD plants, particularly some of the larger groups, and more detailed analyses where you have fossil calibration and can evaluate divergence times. So more of this needs to be done. Also, phylogeography, this is an opportunity to do niche modeling for some of these groups. Some of this has been done. Some of the Carex species have been evaluated with regard to that, and Mackenzie Mabry did do some of this, for example, in the study she did with Cryptantha showed that the shift from that major South American clade in the upper, the blue box, showed a shift from what is currently a Mediterranean climate to what is currently a desert climate in South America. So I think more of these need to be done in terms of evaluating what the areas look like at the time of dispersal, what they look like today, and kind of teasing it apart, particularly if you had multiple populations in both continents. So right now we're more at the coarse scale of things. So a really detailed, more population type study would be great to do in an AAD plant. And some of us, particularly Mackenzie Maber and others, are interested in doing molecular developmental studies to evaluate the molecular basis for some of these changes that we see in plant duration, in polyploidy, in cleistogamy. For example, does the evolution of polyploidy help explain the evolution of cleistogamy and plant duration changes? Do an increased number of sets of chromosomes allow for the evolution of some of them to accommodate, changes in polyploidy and cleistogamy? And what's happening at the molecular level rather, to explain cleistogamy, to explain the shift to from annual to perennial? A lot of this is not known currently, and the AAD model, I think, is ideal for evaluating these changes, these major shifts in plant evolution. So that's a synopsis of our work on AADs and a little bit about Cryptanthas. I have a website that I maintain. You can photograph that QR code if you want, on American Amphitropic disjuncts, where I have these all by different bioregions and a lot of information about it based from our article. So check that out sometime if you're interested. And here's an example of a page where we list all the North American, South American examples, the literature that we get the information, even shifts in chromosome number, and the timing and direction of dispersal. And that's my talk. If you're interested in popcorn flowers, there are the QR codes for our Amsinckiinae website, for the AAD page again, and for our new SDSU Biodiversity Museum. So thank you very much. Okay. It's always kind of weird since I can't hear anybody. Everybody heard me, right?
Thank you, Mike.
Yes.
Really wonderful. So if you have a question, you can raise your hand, or you can also put it in the chat. We'll start with Andy Simpson.
I have two questions.
Yeah.
Number one, there has been a lot of climate change in the last 6 million years. Does this inform the differential timing of the colonization of the bipolar, temperate, and desert distributions?
Yeah, the bipolar is relatively easy since they were all fairly recent in time.
Yeah, I'd imagine the climate hasn't changed nearly as much.
It's really just the receding of the glaciers that has happened. The others, yeah, that's an obvious and interesting question. Mediterranean climates haven't been around that long. And so what they look like 6 million, what did the habitats look like 6 million years ago? Desertification hasn't been around that, in Chile.
There were something resembling Mediterranean climates in Nevada 15 million years ago, based on what was living there at the time. But
okay, all right.
but they probably were different in
Yeah.
ways that we cannot fathom.
So that's a very good question, and something that we need to look at a little more closely, which we haven't yet. That I talked about in general for phylogeography, evaluating what the habitats looked like at the time of dispersal versus what they look like today. Unfortunately, we don't have, for our Cryptantha group, we have we have no fossils of Cryptantha in our clade. what had been thought to be fossils of Cryptantha. 3 fossils turned out to be not Cryptantha at all, another member of the Boraginaceae. So an excellent question. I agree with you. That's work that we need to do for our popcorn flower group and many more of these studies to carefully tease apart what these areas look like. I mean, we're basing it, as you know, on what's there currently, what their current distribution is and what the current bioregions are today, not what they were in the past. So good question. I want you to work on that. Always put it on you. All right. Investigate it further. What was your second question, Andy? Did you have
My second question is the remarks to is remarks to the directionality of most of your groups, apart from the desert plants, colonized from North America to South America. That's a pattern that has been noticed in animals as well across the Great Panamanian Interchange 3 million years ago. Do you think there's a connection, a connection there, a causal link for why both kingdoms would be doing that?
Yeah, in the animal example, I think there are many more. I'm not real familiar with that literature. That's probably in many cases explained as a vicariance event. And some of my colleagues have argued that for plants as well. You know, the other hypothesis is that these plant populations could have had a wide range of distribution and then simply went extinct in the tropical regions. I don't think that's true for the plant examples. We don't have any fossil examples of these AD plants that occur in the tropics. They would not have been preserved anyway, most likely, let alone discovered. I think the bird migratory pattern is the best explanation for plants. I think for some of the animal examples, yes, a migration and a vicariance from north to south. In terms of why from north to south, I don't know. In the animal examples, are you familiar with that literature? I'm just not.
Among mammals, the predominant fauna of South America before the Panamanian Interchange was marsupial.
Right.
Unless you go way, way back, and then there were placental orders that are just straight up extinct.
So I guess the hypothesis is, you know, when the isthmus closed, there was a migration of non-placental mammals to the south and maybe displaced many of the marsupial mammals.
And possums came north.
Yeah, possums were the ones that survived, did quite well.
Yeah.
So I don't know if there's any great correlation between the two. It's an interesting thing to think about. And, you know, the bird migratory patterns, to my knowledge, really haven't been worked out. A student of Bruce Baldwin's, I just contacted Bruce if he's there, had some ideas about this in his thesis, hasn't been published yet. You know, the specific bird migratory patterns, north to south, south to the north, you know, need to be evaluated, I think, a little more carefully. But yeah, good question about the animal examples.
Hey, Mike, if I may jump in, with respect to the disjuncts across the Isthmus of Panama, the huge majority of them are tropical plants and would not be found in the amphitropical disjuncts.
Correct. Yeah, good point. In terms of amphitropic examples, you just don't see that. Yeah.
Peter, did you have a question?
Yeah, thanks very much. So in the cases where amphitropical distribution is due to naturalization, are similar character shifts seen in the recipient populations?
Yeah, I don't think we've had enough time to see that. Very good question. And in fact, I kind of propose that would be an interesting avenue of research to really look, and people are working on this in terms of naturalized plants that have migrated, say, from the Mediterranean region to California, the changes that have occurred after that. In terms of that hasn't really been studied, and I don't know of any shifts that we have examples of the, you know, the major criteria that I mentioned, shift to polyploidy, to the perennial duration, and cleistogamy. I don't know of that any of that has happened. But this whole thing of, you know, changes that occur after long distance dispersal, whether it's by humans or naturally, is really interesting in itself. And I think maybe somebody knows, I think there may have been some examples of shifts in polyploidy. Anybody know that, of a naturalized plant that came to North America? Anybody want to pop in on that? Then I'm not
Tragopogon.
Tragopogon in as an example of
Of a newly formed polyploid in the introduced range.
okay, in an introduced range, but
Yeah.
that would be from the old world to the new world,
Right, not amphitropical.
not amphitropical, but still a long distance migration or dispersal event. Okay, so that's an example then of one that did exhibit the shift after, you say, long distance dispersal that isn't amphitropical, but similar thing. So I think it's an interesting avenue of research. Naturalized plants that we know are naturalized, say in California, the changes that have occurred even in relatively historical times. And I know people are working on this genetic changes. There's a new field called urban evolution. We actually have, that's our most recent hire here. Kinsey Brock, a herpetologist, is looking at changes that occur in lizards as they are introduced into new regions. So that, I think it's wide open for plants, and urbanization is only going to increase, unfortunately. John Redmond's looking at plants in our urban canyons and finding some interesting things. So I don't know where I'm going on this, but I think that the shifts that we see in evolutionary shifts we see in AADs could well happen in introduced plants as well, given time. And they're certainly diverging genetically.
Catherine had a question.
Yes, hi. Thanks. I really enjoyed your talk. I hadn't actually heard of this type of disjunct distribution before, but I have heard a lot about Galapagos species being related to things in the desert Southwest, because that's the group that I work on actually has that type of disjunction. I sort of wonder whether it's not all part of the same process. I know the Galapagos are right in the middle of the tropics. They're on the equator, but it's a desert-like environment, and
Right.
it's kind of a transfer between different deserts, basically. And I sort of wonder if that should be sort of analyzed the same way, or if it's a totally different thing and I'm missing something.
Yeah, good point. It is right near the equator. So in a sense, it's tropical. But I think we included that. Michael, are you familiar with Michael Moore's work on Techilia? If you know the genus Techilia.
Yes, yeah.
In what is down the Aridiaceae, a segregate family of the Boraginaceae.
Oh, wow. I didn't know about that one yet.
Yeah, so
Okay.
Techilia now, I mean, as I and people in our group classified in the Aridiaceae.
Mhm.
And I believe I didn't even think about that, but we included it as an example of an AAD plant. Even though some of them, you're right, occur technically in a tropical region, the environment is desert.
Right. Mhm.
So good, very good point. Didn't even think about that when we were including it.
Yeah, and
That-
we can have the same kind of hand-wavy explanations for how they got to the Galapagos. We're always like birds, even though amaranths have no stickers or anything, they can't attach to anything very easily. We're thinking maybe birds pick up mud and the seeds are in the mud or something, and it's
Sure.
all. I really like to read that Collins master's thesis because I don't really know of any empirical testing for that, but
Yeah, contact me if anybody wants a copy of it. I have a PDF. I could
awesome. Yeah.
be great. But very good point. Something I hadn't really thought about, I have to admit, the Galapagos, the Techilia examples, we included them as examples of AADs. I think some of them occur further south, more into the amphitropic zone. I'll have to look at that. But I'll look at I'll send you Michael Moore's work on Techilia as well.
Cool,
Good point.
thank you.
Bruce Baldwin. He had his hand raised.
Bruce, you there? You have any insight about bird migratory patterns? We
Oh,
can't.
no. His hand went down. I'm not sure.
Did okay. I don't see him.
Well, Nancy had a good question in the chat. She was asking whether species that have dispersed one way or the other have stickier seeds than other species. Like, are
Oh,
there
yeah.
other morphological traits?
Yeah, that's been studied quite a bit. In fact, Collins evaluated that, and they do. When you compare them, some of them have very sticky seeds, either gelatinous seeds or a lot of them, like, many of the Cryptanthas have accessory tissue that's bristly. Some members of Acina of the Rosaceae. I remember walking through that, which is an example in Chile, and it's all over your pants when you walk. There's one of those stick-tight plants. They do indeed. And there have been articles written on that. In fact, Matt Williams evaluated that for our popcorn flowers and showed that the ones that did exhibit the AAD pattern, two things, the netlets tended to be smaller and rougher. And, I don't know if that has a whole lot to do with it. It may be correlated with the pattern, but I think probably the calyx surrounding the nutlets is maybe a more important dispersal mechanism. So yes, that's definitely true. They tend to be stickier in one way or another.
Okay.
Hey, sorry,
Bruce
I
is
couldn't get
on
my
now.
hand or couldn't unmute myself there. I wanted to know whether I wanted to stay in the meeting or not first, which was a strange question. But yeah, I just want to follow up on Mike Park's results with the
Please do.
amphitropic dispersal with California to Chile. And what he found was looking at some of the more the migratory birds that make that trip regularly was that they tend to go from California to Chile, but when they're heading north again, they head right up through the center of the continent in a race to get to their breeding grounds in the northern hemisphere. It's quicker to just go north and not go back along the coast. I mean, that was true for the knot, I think, and I can't remember all the others, but it's quite a simple explanation for why the dispersals tend to go from north to south with regard to California floristic province down to Mediterranean Chile. Yeah, very elegant, but it's chapter 3 of his unpublished dissertation, but it is filed at Berkeley, so it's published in a sense there, and hopefully he will get it out at some point. But I think he spoke about it, didn't he, Mike, in the same symposium that you talked about?
Yes, it's true. He was going to write an article as well, but didn't happen. But
yeah, anyways, it's quite a nice study, and he had more data on the migratory pathways he wanted to incorporate into a revision of the chapter, and that's been the bottleneck in getting it done.
very good. Bruce, I don't recall going from south to north in that example from Chile. Is it still a single flight? In other words, one idea is that some birds may make more frequent stops, and
well, in that particular pattern, the Mediterranean pattern, the flight north is not back to California.
Right.
Whereas the desert pattern is different, and that involves different birds, presumably, or it's not the same group of players. Yeah, that's about all I can say about it at this point.
Good. I'd forgotten.
Been a long time ago, over a decade ago. I'm a little fuzzy on the details at this point.
Well, try to get that work published if you can.
Yeah, I mean, it's Mike's to publish. I definitely wouldn't try to publish it on my own, but
Very good.
yeah, well, hopefully that can be published before too long, much longer.
Jerry, did
I
you
have a question. Hi. Hello, Bruce.
Hey, Jerry, good to see you.
Yeah, question for Mike. First of all, thanks so much for all of your work on that very easy taxonomically Easy family.
Pretty good. I find poplar flowers easier than some groups.
Well, also, I have a kind of a specific question. This is a very fascinating topic, and I remember reading about it years ago when it was, I can't say his name now. This happens to me all the time. Former director at Missouri Botanical Garden, Peter Raven. There you go. I got it. Ranunculus bonariensis. Are you familiar with that as a potential example of this AAD? It's in California, and we found it just in 2017, a couple of sites in Oregon, in southern Oregon, the nearest site being a couple 100 miles south in California.
Okay.
And the Jepson 2, it's mentioned that it's in South America. And there's no mention of it in Mexico, so it would seem that it might be one of these examples.
It is an example. If I can share my screen, here is the website that I mentioned. There it is. See it, Ranunculus bonariensis?
Yeah, right.
So, and the green there means that that same species. Right now, we're listed as a variety Tricephalus.
That's what the Jepson manual has it as that.
Occurs in both continents. So we have the GBIF maps linked to this, so you can see its distribution. And
good, great.
Screen right there. So yeah, that's one of our examples. I'd forgotten which ranunculus it was.
Yeah, it was kind of a neat find to get it up into southern Oregon from otherwise just northern California. So
Nice.
But again, thanks for the borage work.
Oh, you bet.
It's still beyond my comprehension, but you've helped a lot.
Another sequence analysis, which we'll be finishing up in the next month or two. Some of these interesting taxa, cleaning it up a little bit. But that story of that Meridime clade. But if anybody out there knows of an example of an AD plant that isn't in our paper, isn't on the website, please contact me. I know a few that have been published recently, but I'd like to begin to revise that in an article. It's a fascinating topic, so I want.
Oh, it is. And thanks for a comprehensive treatment of it.
I want all of you.
Such a fascinating topic. I feel like people could ask you questions all night long.
That's true.
But we're already well past 8, so maybe it's time to say goodnight. Thank you so much for your wonderful talk. It was really fascinating. And yeah, we'll post a recording of this on our website, so anyone who didn't get to see can watch it later.
And if you can save the chat for me, I'll take a look at some of the comments and questions. I'll try to save it myself, if you can do that.
Okay.
Well,
anyway. It might be beyond my Zoom skills. Mitchell, is that something you know how to do?
Yes, we can definitely do that. No problem.
Excellent.
I'll send when we post the talk on our YouTube page, I'll send you the link and then a copy of the chat.
Very good. Thanks, Mitchell. Very good.
No problem.
All right. Good.
Great talk. Thank you so much.
Good night, everybody.
Good night, everybody.
Thanks for coming.